My name is Emily Ball, and through the Moffitt Spark program, I had the opportunity to work in Dr. Alexander's lab, where we study the CMG helicase as a potential cancer therapeutic target. As I said, we study the replicative helicase, and during S phase, the phase at which DNA is replicated, the helicase, as you can see right here, works as a zipper to unwind the DNA and to initiate replication. This allows the DNA polymerase to come in and synthesize a new strand behind it. The current standard for care for cancer patients includes many chemotherapy drugs, many of which prevent replication by causing stress during this S phase. However, no drugs have yet been developed that target the helicase specifically. By studying this enzyme, we aim to provide new, novel drug targets. The active form of this enzyme is made up of 11 subunits, collectively called the CMG helicase. The M in the CMG helicase stands for the MCM complex. This is a 6-membered ring, which serves as the ATPase core of the enzyme. The G stands for the GINS complex. It is made up of 4 subunits and is involved in helicase stability. And the C stands for CDC45, which is an MCM cofactor that has been shown by our lab to be extremely rate limiting for helicase activation. MCMs are first loaded before replication. Then certain MCM complexes are shown to become active, CMGs, upon recruitment of the GINS and CDC45 complexes, which increase the helicase activity of the MCMs by about 300-fold. The CMG pairs work in opposite directions to separate the DNA strands and create replicative forks. In this cell, the DNA is not linear as is shown here in this picture. It's actually condensed and coiled upon itself to create a larger, denser chromatin structure. In order to unzip the DNA, the chromatin must first be decondensed. Much like in order to separate 2 strings of yarn, you need to unwind the string from the spool. Therefore, chromatin decondensation must occur before or coincide with helicase activity. Our lab and others have shown that CDC45 plays a role in chromatin decondensation at the replicative forks. However, little is known about the mechanisms and other players involved. The experimental approach was originally used to show that CDC45 can open chromatin was a chromatin unfolding assay using Chinese hamster cells, or CHO cells. The CHO cell genomes were modified to include a homogeneous staining region, which we call an HSR. These regions contain LAC-O binding sites, which serve as a binding counterpart to LAC-I. These LAC-O-LAC-I binding interactions can be visualized with a fluorescent anti-LAC-I antibody, which will appear as closed dots representing condensed chromatin. If a gene is capable of decondensing chromatin, you target the gene to these HR regions using the LAC-I tag, then the HSRs will appear as an open structure, as is seen with CDC45. Here you can see examples of these closed and open forms. When the LAC-I only was transfected into the cells, the HSRs appear as closed dots, as you can see in this top picture right here. However, when LAC-I CDC45 was transfected into the cell, many HSRs appear as open structures, as you can see in these bottom 2 pictures, and larger in this final picture. This summer, I worked to verify this data. Using the same procedure as before, CHO cells were transfected with LAC-I tag CDC45, or the mutant version of LAC-I CDC45, which lacks the C terminus. A Western blot was used to verify the expression, and immunofluorescence was used to visualize HSRs. When the wild-type CDC45 was overexpressed, more HSRs were observed and the total number of cells counted, as you can see with this dark bar. So the CDC45 wild-type causes more open HSRs, whereas the CDC45 mutant, more closed HSRs were observed in the total counts. This information suggests that when wild-type CDC45 was overexpressed, more open HSRs were observed in the total number, and this suggests that CDC45, the C terminus of this molecule, is responsible for this opening, or is interacting with other proteins that cause the decondensation. Information about how CDC45 plays a role in modifying chromatin could lead to the development of new therapies to target helicase activation and inhibit replication, either by targeting CDC45 itself or the activity of a protein associated with this function. Therefore, in the future, we plan to investigate a variety of different players that could be involved, starting with the histone acetyltransferases, or HATs for short. This week I began to explore these by co-transfecting hats TIP60 and GCN5 in their mutant versions. I'd like to acknowledge my mentor, Dr. Mark Alexandro, and my lab mentor, Brooke and Victoria, also the Moffitt Cancer Center, the SPARK program, and all of our sponsors. Thank you for your attention. If you have any questions.
Questions? Is it known which of the other proteins is the primary target of CC45?
Not yet. No, that's one of the things we're investigating. We're starting with hats first, and we're specifically TIP60 and GCN5.
And why those?
Well, hats are known to decondense chromatin, and that's just our starting point. A little bit of fishing.
Fair enough. Fisherman catch fish. Other questions?
Thank you very much.